Yeah, thanks, Ben. Sorry, I'm not as pretty as Alex, but I'll do my best to give a talk that's at least as interesting. I am, of course, the Chief Science Officer of a foundation that works on a large number of aspects of aging, but the area that I first got into 15 years ago was the role of mitochondrial mutations in aging, which is very much what Alex would have spoken about. So I hope this talk is going to be an adequate replacement for the one that you're not going to hear. Let me see if I can move this a little closer. There we go. Right then, so for those of you who are completely new to the whole concept of mitochondrial mutations, let me start at the very beginning with what DNA we're talking about. This is the mitochondrial DNA. It's extremely small. It's only 16.5 kilobases. And it's the only DNA that mammalian species have outside the nucleus. It encodes just 13 proteins, which are all essential components of the respiratory chain, the system that utilizes oxygen to extract energy from nutrients. And these 13 proteins are very well conserved. It's actually pretty much, without exception, throughout the animal kingdom, exactly the same 13 proteins encoded in the mitochondrial DNA. And you'll see why. It just turns out that transfer of proteins between the mitochondrial DNA and the nucleus is a tricky business. It used not to be a tricky business. The mitochondrion came into existence a couple of billion years ago by what's called an endosymbiotic event, the engulfing of a bacterium by another bacterium of a different sort from the archaea. And at that time, the bacterium that was engulfed had upwards of 1,000 genes, at least. Many of those genes were just lost subsequently, but a lot of them were transferred spontaneously to the nuclear DNA. And there has been some debate over the years about why this process did not run to completion, leaving the mitochondrion without any DNA at all. I'll be coming back to some of the reasons for that in a second. Now, the history of all of this is worth summarizing a little bit. The fact that the mitochondrion has DNA at all was only discovered in the late 1960s, and it was just a few years later that the famous Harman, who in the mid-50s came up with the idea that free radicals might be important in aging, suggested that the mitochondrial DNA might actually be the mediator of all of this. That the mitochondrial DNA might, first of all, be the main victim, the main important victim of free radical damage, because it's in the wrong place. It's right next to the respiratory chain. A few years after that, the famous and eminent and subsequently much more famous for other reasons gerontologist Alex Comfort noticed what Harman had said and suggested that it couldn't possibly be right, because he was aware of something that until quite recently many people did not really pay much attention to, namely that in non-dividing cells, as well as, of course, in dividing cells, there is constant mitochondrial replication. Mitochondria are constantly being created. And of course, in non-dividing cells, that process has to be balanced by the elimination of other mitochondria. And Comfort said, well, obviously the mitochondria that are going to be destroyed are the damaged ones, so damage cannot accumulate in mitochondria during aging, except perhaps as some secondary consequence of some other primary driver of aging. And that was pretty much that. There was almost nothing. If you go back to the literature throughout the late 1980s, you'll find basically nothing about mitochondrial mutations until roughly 1989, when an Australian mitochondriologist named Tony Linnane blew the lid off all of this again by having a proper look at the problem and figuring out that actually quite a lot of mitochondrial mutations could be seen in older organisms relative to younger organisms. And this became a big debate very quickly. A lot of people said, well, okay, we see an accumulation of mitochondrial mutations during aging, but actually the absolute amount, the absolute proportion of mutant mitochondria, even in elderly organisms, is still pretty low, therefore it probably doesn't matter. And some other people said, no, it probably doesn't matter, it's just a bit subtler, and so on. It became a nice row. This was Harman's vision. This is actually a quote from the paper in 1972 that he wrote. You know, it's really, to my mind, actually an even more significant contribution to the progression of gerontology than his original work on the free radicals, because he was one of a number of researchers who were thinking along more or less the same lines with regard to the role of free radicals in aging back in the 1950s, and if he hadn't published what he did, then someone else would have come along pretty soon afterwards. Whereas, as I say, in this case, he was, well, more or less 17 years ahead of his time, and it's rather a shame that people didn't take any more notice of his insight earlier on. Now, this is where things started to get interesting. In the early 1990s, especially, and actually there were some hints of this even in the 80s in disease models, it became apparent that the age-related accumulation of mitochondrial mutations is not uniform across tissues. That one typically sees a mosaic accumulation, where some cells are really just taken over completely by mutant mitochondrial DNA, whereas most cells are almost, if not completely, devoid of such mutations. This is what you see if you ask that question using histochemistry. This is simply a transverse section through skeletal muscle looking with a stain for cytochrome C oxidase, one of the components of the respiratory chain that's partly encoded in the mitochondrial DNA. And as you can see, there are plenty of these muscle fibers that stain heavily, and there are others that don't stain at all. This is what you tend to see in such circumstances. The only unusual thing about this photograph is that you've actually got two fibers fairly close together that are cytochrome oxidase negative. This is basically what was wrong with the argument that Comfort made to reject the possibility that mitochondrial mutations could matter in aging. He just assumed that damaged mitochondria would be eliminated. And he's right in a way, as I will describe later on. But it turns out that, at least some of the time, mutant mitochondria are not eliminated. That type of damage is not detected in the appropriate way by the machinery that eliminates mitochondria. And in fact, the opposite happens. The mosaic distribution of mitochondrial activity that I just showed you, when you start to look at it at the level of DNA, turns out to come out like this. This is a southern blot from a paper published in the early days by Doug Turnbull's group up in Newcastle. And what you're seeing here is the is how one particular restriction fragment from the mitochondrial DNA looks at the level of single cells. What you're seeing is that in most cells, this particular restriction fragment is of this length, and that's all you see. You just see one particular band. But there are some cells where you just don't have that band, because, as I say, they've been taken over by mitochondrial mutations. Now, the thing that this sort of experiment shows you that the histochemistry I showed you earlier does not, is it shows you that it's not just that the cell has been taken over by a lot of different mutations and therefore there's got no wild type. The cells have been taken over by just one mutation. You've just got one other band, down here in this case, down here in this case, here in this case, where you've still got a small residue of wild type. This means that it's not a cascade. It's not what some people used to call a vicious cycle of mutations causing increased free radical production, causing elevated mutation rate, and so on and so forth. What you've got instead is a clonal expansion of a very rare event, a mutational event, that somehow enjoys a selective advantage thereafter. And my first paper in this, in gerontology, back in '97, proposed a mechanism for this, which essentially said that the reason why this happens is because mutant mitochondria don't damage their own membranes as rapidly as normal mitochondria do. Mitochondria, of course, by virtue of their respiratory activity, create free radicals. The free radicals damage DNA, but they also damage lipids and proteins. And I hypothesized that maybe this is how mitochondria that are mutant clonally expand, that they don't actually get replicated any more readily than normal mitochondria, but they get destroyed less readily than normal mitochondria. It's taken a very long time for any real evidence either way to come out with relation to this hypothesis, but just in the past few years things have started to come together and it's looking rather good for this hypothesis. First of all, something that wasn't really understood at all 15 years ago is that there are two mechanisms for the elimination of large things that the cell doesn't want, a process called macroautophagy. These two alternatives are called bulk autophagy and selective autophagy. Bulk autophagy happens when the cell just needs raw materials, amino acids and stuff like that. We're going in a starvation scenario. Selective autophagy is an ongoing maintenance mode situation. And the mechanism for selective autophagy of mitochondria, mitophagy as it's often called now, has now been reasonably well elucidated. There's a particular protein that is made constantly in the nucleus, in the cytosol, and goes into the outer membrane of mitochondria. And in mitochondria that are working well, that protein is imported into the inner membrane and broken down. In mitochondria that are working less well, the protein remains in the outer membrane and sequesters a second protein, which causes ubiquitination of other proteins. And I'm not going to go into the details, but the long and short of it is we now pretty much understand how selective mitophagy works. And it turns out that mitochondria which are really broken, they can't make any kind of membrane potential because they have leaky membranes, for example, are broken down this way. But mitochondria that are only mutants, but their membranes are intact, make enough of a membrane potential so that this mechanism is not activated. So it's looking pretty good for the hypothesis, but we still have plenty to do before it can be really said to be confirmed. Now, a very interesting thing that also happened in the past decade or so in this area was the discovery of new cell types in which this phenomenon occurs. It was originally thought to have been established that this phenomenon of clonal expansion and mitochondrial mutations happens only in post-mitotic cells, or at least in very slowly dividing cells. It was found in skeletal muscle, it was found in the heart, it was found in neurons, and that was pretty much it. It was found in a few other long-lived cell types. People really didn't think that that was all that surprising, because in regularly dividing cells, you've got completely different population dynamics, different things driving the whole mitochondrial population, in particularly whenever a cell divides, the requirement to have that mitochondria divide too so that you don't run out of mitochondria. But then about 10 years ago, Tom Kirkwood and Doug Turnbull discovered the most extraordinary thing, that actually mutant mitochondria also accumulate in the stem cells of the gut epithelium. And this turned out to be a really useful phenomenon. You can tell a lot about what the lineage of cells in the gut are actually doing by staining for mitochondrial respiratory chain activity. These are the sorts of photographs that you'll see if you look at literature from that group and from other groups. Malcolm Allison's group in London started to look at other tissues in this same sort of way, and were able to establish that the phenomenon is actually more widespread. This is from the liver, I believe this one's from the pancreas. You can find this in a lot of areas, and it's turned out to be a very useful tool. But the big question is, does it matter? And if Alex had been here, this is the sort of work that she would have spoken about. Alex's group are one of 3 or 4 groups around the world that have been looking for a number of years now at a very interesting mutant mouse model, which has a deficiency in the proofreading function of the DNA polymerase that replicates mitochondrial DNA. There's a special polymerase gamma, and this particular mutation accumulates mutations in the mitochondrial DNA, even though the polymer itself is a nuclear gene, I hasten to add. It accumulates mutations much more rapidly than normal. And these mice, they get pretty unhappy pretty quickly. They have a number of phenotypes that one could regard as proteoric. You can see what sort of trouble they get into right here. You know, they've got kyphosis, they've got various problems. In particular, they've got some rather curious problems that one wouldn't call typical of accelerated aging. They get a lot of anemia and alopecia and so on before getting problems in their post-mitotic cells. And some people have pointed out that this actually is a bit of a problem for the model, because really we want a model that recapitulates the cell type distribution of the mitochondrial mutation accumulation seen in normal aging. Another group in Europe, in Finland actually, the group of Anni Suomalainen, have developed a different mouse model, a dominant negative mutant in a helicase named Twinkle, which is important in DNA replication. And these mutants seem to be somewhat better at recapitulating but accelerating normal aging, in the sense that, first of all, they accumulate the right sort of mutation. Mutations in normal aging in mammals in general tend to be mostly deletions, where a large chunk of the molecule just goes missing. Whereas the mutations that accumulate in these polygamma mice tend to be almost always point mutations. That, you might think that's not a very important difference. The twinkle mutations are deletions, basically. But it might be an important difference because it also extends to the cell types. If you look at where things are going wrong initially in these polygamma mutant mice, you find that it's mainly in the stem cells of these compartments that give rise to rapidly renewing tissues. So the skin, which is why we're looking at alopecia, the blood, why we're looking at anemia. You know, it's not really very typical of normal aging. So this model is valuable, that Alex was spoken about. The model from Annie's Alliance group is also valuable. My personal hunch is that Annie's model is going to be more useful in the end, but both of these models need more work and more refinement to make them more faithful recapitulations of normal aging. So at this point, we don't really know whether mitochondrial mutation accumulation actually matters in normal aging. So there was a brief mention earlier of my 7-point plan for you know, the types of damage that exist in aging and what we need to do to fix them. And I think it's fair to say that mitochondrial mutations are a bit of a Cinderella among those 7. For all the other 6, we can look at a particular major cause of death in humans in the Western world, and we can say that it's mainly driven by that particular type of damage. Whereas, in the case of mitochondrial mutations, we cannot definitively point to any such pathology. However, that's not to say it's not true. It may very well be that mitochondrial mutations have a very ubiquitous, albeit subtle and elaborate role in a wide range of different aspects of aging. So ultimately, we're not going to know until we fix the problem. I'm sure many of you have thought and read about the ultimate, you know, philosophical difficulties of deducing information about normal aging from models of accelerated aging. Essentially, one doesn't know whether one's accelerated it so much that one is you know, disrupting the system in a manner that's not informative. There are various other problems with accelerated aging models. What one really wants is a decelerated aging model. And so this is the question: can we actually fix this problem? And one alternative is to try to eliminate the cells that are problematic. But this turns out to be a lot harder than it sounds, especially because skeletal muscle is affected very badly in this respect. And what happens in skeletal muscle is you don't lose mitochondrial function in the whole fibre, only in a small segment of it, and it would be a bad thing to be killing the whole fibre because by old age most fibres have some segment somewhere in them that is mutant. So then the question is, you know, can we in some way make these mutant cells harmless? Can we somehow rescue the problem? And that's what we'd like to do. In the lab, in Science Foundation's lab out in California, one of the big projects we're working on is something that was first proposed back in the mid-1980s, but no one's really got it working yet. Essentially to put the mutant mitochondrial DNA into the nucleus to finish that job that evolution had left unfinished of moving those genes from the mitochondrial DNA. The way that we propose to do that is, first of all, to do very straightforward point mutations that will compensate for the differences of genetic code between the two gene ends. Secondly, to add an N-terminal leader sequence so that the protein is recognized by the cell as being destined for the mitochondrion. However, there's a third problem, and this is generally believed to be the main reason why evolution has not completed the job of moving the DNA. Namely that these proteins are very hydrophobic, and the protein import machinery that takes more than 1,000 nuclear-coded proteins and already naturally imports them into the mitochondrion is not good at handling really hydrophobic proteins. People have been grappling with this problem for a long time. Back in the early 2000s, about 2 minutes, right? Back in the early 2000s, a group in Bethesda in the NIH seems to have got lucky and got one of these genes working, but nobody's really figured out why it worked, so we can't really use that information. However, a few years ago, a group in Paris did something which was much more in the way of rational design of a way around the hydrophobicity problem, essentially figuring out how to get these proteins to be imported while they are still being synthesized, and therefore to stop them from folding up and from letting their hydrophobicity hinder import. There's another rather clever idea, which I'm not going to say much about because Carla Kerr is apparently here and going to give a talk tomorrow about this. But suffice to say that we might be able to solve the same problem, at least in the case of certain types of mutation, by importing not proteins but RNA. And these are some figures from a paper that Vicky and her colleague Max Keitel published recently, showing essentially that you have to do 3 particular things to these RNAs to get the whole thing to work. And if you do any subset of those 3, it's completely ineffective. So I won't tell you what the 3 are because I don't want to steal Carla's thunder. But the long and short of it is, this is a very important area of translational gerontology. We don't know that mitochondrial mutations matter in aging, but if we fix everything else that we do know matters and we all carry on dying on schedule, we're going to feel pretty stupid that we didn't actually try and fix this problem too. So I take the view that we should get on and do our best to fix it. Allotopic expression seems like an interesting way to go. It seems like it's got promise. We still don't know whether we can get it working, but as I've just outlined, there are a couple of alternative ways that we might get it working. And I hope you're interested in paying attention to this research as it moves forward. Thanks very much.